replication is a key process for cellular life, as DNA will replicate and copy itself to produce two identical daughter strands from the original DNA strand in the nucleus of a cell for eukaryotes. DNA is a polymer made of many building blocks called nucleotides in a double helix structure. There are three components of a nucleotide: a deoxyribose sugar, a phosphate group which links neighbouring sugars, forming the backbone of DNA, and four different nitrogenous bases. These include adenine, thymine, guanine, and cytosine. To ensure DNA is double-stranded, the four nitrogenous bases are complementary paired with another base. For example, adenine and thymine are complementary to each other, thus forming a double hydrogen bond, whereas guanine and cytosine undergo complementary base pairing. Three hydrogen bonds are formed. This bonding ultimately leads to the formation of a functional double-stranded DNA molecule. A and G are both purines, meaning that they will always pair with pyrimidines, T and C, in the opposite strand. DNA is semi-conservative, meaning that one parent strand becomes a part of the daughter strand. The synthesis of DNA occurs and builds in a specific direction from 5' to 3', although the other complementary strand of DNA runs 3' to 5'. To ensure replication of DNA goes ahead, multiple enzymes are used. To begin with, an enzyme called helicase will insert itself into the double-stranded DNA and unzip and unwind two strands by breaking the complementary bonds. It usually starts at areas rich in A-T bonds, as the two bases only share two hydrogen bonds, thus easier to break. Helicase will stop once a large enough origin of replication is formed. After separation, the helix destabilizing proteins, SSB, bind to the unzipped DNA strands to inhibit the reformation of the two strands. Consequently, another enzyme will be used to stop the DNA from supercoiling, to stop it from compacting. Step 2 in DNA synthesis is priming the parent strands. Another enzyme called RNA primase is the initiation point of the new daughter strands, attracting complementary RNA nucleotides to the free DNA bases to bind. Essentially, RNA nucleotides are the primers for the binding of DNA nucleotides, signaling for DNA polymerase III where to begin building. Next is elongation. This process occurs differently between the two parent strands. This is due to the leading strand beginning at 5' through to 3', where the lagging strand is opposite, 3' to 5', making it anti-parallel. This causes one strand to be leading, allowing continuous synthesizing, and one lagging where it's discontinuous synthesizing. To aid this essential process, enzyme DNA polymerase builds the new daughter DNA strand, but can only build from 3' to 5'. DNA polymerase III will begin to find DNA nucleotides in the nucleus and begin binding with their complementary base where the RNA primase has started the new strand. The sugar and phosphate backbone are also formed when DNA polymerase III builds the strand. For example, if the leading strand has bases A, C, T, G, then DNA polymerase III will begin where the RNA primer has begun, adding complementary nucleotides T, T, G, A, C. This ultimately leads to the development of the leading daughter cell. Because DNA polymerase III can only build in 5' to 3' direction, it has to race and backtrack to where the unwinding is occurring. This leads to the lagging strand. This lagging strand, RNA primers have to continuously be placed back upon themselves onto the original strand to allow DNA polymerase III to bind DNA nucleotides, producing little fragments of DNA called Okazaki fragments. The primers have to be replaced with DNA bases, since the primers are RNA nucleotides. To glue these fragments together, the enzyme DNA ligase will glue the gaps between the Okazaki fragments, sealing them into one long strand. It's important that DNA replication produces the identical daughter strand to the parent DNA molecules. For example, if one of the nucleotides are matched to the wrong base, it is possible that it will encode for the incorrect gene, which can result in incorrectly coded protein or no protein at all, affecting the function of the cell. DNA polymerase I, or exonuclease, will read back through the strand and edit out mutations and mistakes that might have occurred and substitute the RNA primers for DNA nucleotides between the sections that have undergone elongation. DNA polymerase II is also a repairing and editing enzyme as it is a backup for DNA polymerase I and III. At the end of this process, two identical DNA molecules are produced. This is called semi-conservative, as the two copies of DNA have one older original strand and one newer strand.